Right. That's my title. So we'll move on to, is it moving now?
Yes, we've got the next slide.
Right, okay. Setting the scene. You can read the notes that are there. The only thing I would add here is the UK distribution is restricted, as you can see, to the south and west. And as far as I can find out, only Ludlow Castle has exceeded this range as a breeding site in the past. And I wonder if it's possible that any of these North Wales colonies can ever produce a viable maternity colony. I'll explain that shortly. If you look at its international distribution, you will see that we are the most northerly part of the range. So it's a miracle that they're here, and that miracle is due to our lovely British weather with constant arrival of moist, warm weather from the Atlantic. Worldwide, when you get to a latitude of 53 degrees north, you've only got about 6 hours of darkness. And that is a big problem for female greater horseshoe bats when they're lactating, if they give birth round about the 21st of June, which is what they sometimes do. They have to forage 3 times a night at that time, and that total period can take 5 hours. So we've got a bat that's trying to do 3-period, 5-hour foraging in a 6-hour darkness period. So it is extremely stressful. The climate has to be wet summers. They can't stand arid conditions. If you look at the distribution, they disappear in heavily arid conditions. They need moist summers to keep their insects going. And in this country, they need mild winters for the males and the young to get through their winters. They've got to have the proper roost choices to have, the foraging habitats, commuting, and so on. And if you put all these together, there's a rule called the laws of limiting factors, which says that if all factors are present, you will get the maximum populations of your animals. If you take away one of them or reduce one of them, that factor will be crucial. And that's what seems to happen. And dietary factors are one of the major aspects. I won't go through the whole list here because you can do that at your leisure at another time. All the red papers are focused on diet content. Jones, Duverger, and Ransome 1995 linked aspects of Duverger's radio tracking PhD study with my diet and pup growth data to show the conservation needs. Rossiter's study involved radio tracking females from 2 successful matrilines, and the 2 used 2 separate areas, as we will see later on. Ransome 1997b showed that foraging success level of individual bats does change even in the summer. So summer isn't always a good time, even for the females. Cold will have effect upon them. Now, this is Woodchester Mansion, one of the, I always say, the largest bat box in Britain. It used to be entirely occupied by bats, but it is now run by Woodchester Mansion Trust. That happened in 1989. And the National Trust owns most of the valley. And there is some very good habitat restoration in progress. And our numbers have, in recent years, actually almost reached the figures they were when I started looking at the site in 1959. So there has been a recovery there. Now, I live about 6 miles from the mansion, and that means I can easily visit to carry out monitoring and scientific studies. The only people who live in the mansion is a custodian and his partner, who occupies the flat. I don't know whether you can see. This is where the flat is, and these are where the bats are. If you can see that. Let's go back. That's the background to the mansion. Before the Mansion Trust took over, there were lots and lots of roosts scattered throughout the whole building, and it was essential for the Mansion Trust to operate to remove the bats from the entire building and get them into some cozy area. A section at the back of the building was used. It was attached to a brewery, but unfortunately there were no beer supplies when we were there. It was never used. But the brewery area is exclusively restricted for the bats. There's a Section 106 agreement made, and we fitted incubators. I was the first to use incubators, and I did that in 1994, partly because there was a very big population crash throughout the whole of the UK in the early 1980s as a result of volcanic effects, a whole series of bad winters and terrible springs. The populations had dropped to a total of 20 live births. And that happened in the Forest of Dean and also in Southwest Wales. So they all went down to this very low level, and it was taking ages for them to pick up. So we used the incubators to get the bats out of all the other areas into 2 colonies, greaters in one attic and lessers in the other. It's easy to collect the droppings because they're concentrated there, and you can see that they were being regularly collected all the time. This is what we do with the droppings. We bring them back and weigh the whole sample wet. We then dried 2 200-gram samples to find out what the total dry weight is. We keep a small set of samples in small pots to then analyse for dietary studies later on. What I do is use 12 pellets per sample into a spotting tile like this, and you can tell I'm a bat because I'm upside down. And the droppings, when you put them in the separate containers, are single droppings from random bats that were hanging above it. And you'll get a sample which is actually acceptable for statistical analysis. Gareth Jones did the first studies and he used 20, but I found that I could get away with 12, and this helped reduce the enormous amount of work involved in doing these samples. You have to use sodium hydroxide to make dissolve all the organic matter, which is why you get this lovely brown soup you can see. It makes it perfectly safe. There are no nasties in it, zoonoses, whatever viruses. The whole lot is safe to handle. And what you do then is spread these droppings onto microscope slides. You make the samples look as if they spread out in a square way, and I put 2 pellets on one slide, so I end up with 6 slides for that one sample. And this is what you see when you look down a binocular microscope at a bat that's been feeding on aphodious beetles in the summer. And here we've got one of the mites, the parasitic mites, which live on the beetles. And you've got large fragments of exoskeleton, which allows you to confirm the identification. The identification can only be made down to family in the case of moths, genus in the case of aphodious, and geotrupes, but in the case of maybugs, since there's only one in the country, you can actually know it's that exact species. I followed Jones as procedures and used his sampling technique, which was to collect samples every week, but only sample 13 in the year. We did the last week of 1 month, and you followed it by the first week of the next month, so that this allowed short-term changes to be seen as well as longer ones in the diet. And then you put all of your findings into a notebook. Computers are very good, but they don't do the stuff for you. What we've got here is a page in my feces analysis book, which shows you data for April 2011. If you look at the top, 5th of April to the 24th of April, because we don't really start counting there until the 3rd week of April. And what we've got is a whole series of numbers. On the left-hand side is numbers 1 to 12, which are the slides. In the top left-hand, we've got 100 OOF, and 100 means that 100% was Ophion sp. The Ophion they are wasps, parasitic wasps, and they fly mainly through the winter, but also in the spring as well. Anything which is color-coded was eaten exclusively by the bat at that time. The top one is 100, the next one's 99, the next one's 95. And I believe that that is the result of contamination rather than the fact that it was actually eating several different sorts of insects. So some bats are out eating just Ophion. Others were eating just Melalontha. Mel is short for Melalontha. That's the maybug or cockchafer. And they were eating just cockchafers, and others Ophion. And down you go to blue, which is tipulid, and brown, which is suitable for dung flies. Dung flies are the yellow flies that you see sitting on cowpats, and they are, all these prey were eaten over a 3-week period. And there would have been days when possibly the whole colony was eating Ophion, and other days when they were all out starting to eat nothing but cockchafers. So this is the problem with doing collections of droppings over a period of time. It's better to do the individuals, as we will see later. If you move on to the second column of data, Melalontha and moths now dominate. I would say that the first lot were bats which were stressed on occasions. That's particularly true if you see dung flies. Dung flies are the prey of last resort for this species, although lessers do love them. You get into May, and within the last week of April and the first week of May, you've gone almost entirely over to moths, and there's the appearance of brown trichoptera, caddisflies, but otherwise they're feeding on improved diet, and by the time you get to the end of May, it's 100% moths with 100% of something I couldn't identify. And what you do is you work all this out and you get yourself a pie chart. That pie chart shows you what the bats are eating. I will now move on to my notes. This was the results just for one summer. 1995 was a very hot summer. There was a drought from June to late August. This seems to have favoured moths. The pie chart shows that moths dominated the diet, and aphodious dung beetles, the grey-blue ones, were next. They were followed by tipulids in dark blue, Melalantha in purple, deep purple, and Ophion wasps. Normally in wet summers, aphodious would be more dominant and moths rather less so. But it's quite clear that you need grazing cattle to generate large numbers of aphodious for young bats to feed on in August. And the way in which the diet switches when the young start to feed is something I published some years ago. To come to the research reports, much of the data from this study was commissioned by for English Nature research reports by Tony Mitchell-Jones in the early 1990s. That was a time when I left teaching, just in time to save my sanity. He needed the scientific evidence for setting up agri-environmental payments for landowners under the Species Recovery Programme. So he set me a remit to do the initial study, which had to take Gareth Jones' study into a different level and look at what was going on in a range of colonies to make sure that we could see whether they were the same at all these different sites. The second one was an extension of that, where we were looking at trying to see what we could do to boost the populations. And I did a big analysis on monitoring protocols, which later on became part of the National Bat Monitoring Programme system. We identified key habitat features. The roost sustenance zone was a term that we coined between us. It's since been annexed and renamed by other groups, but that's where it started. And we recommended the land management improvements for each site. It ended up with a leaflet, Managing Landscapes for the Great Horseshoe Bat, and I'm sure no one here would need an introduction to this, because if you're in the Devon project, you would have used this leaflet many times. I'm sure David Appleton would agree with me on that one. Strangely, Anon wrote it. Now, I don't know Anon, but he certainly didn't give me credit for my all the material that was in it. And neither did the Guide for Land Managements, which came out by the JNC, which took most of what was in that leaflet and converted it into a rather amusing version of it. So again, we were strangely ignored. Right, we will leave that and move on to another aspect now, which is the individual diets. My research project involves catching and ringing pups annually soon after birth. Once births cease, we catch the whole colony early on one morning in July. Many are paired up with their mothers. This year, 80% were like this. This allows us to trace matrilines over many generations. Since 1993, all bats have been DNA sampled to fill in gaps in pedigrees. The work started by Steve Rossiter and continues now with Luke Romaine. Morning, Luke. I hope you're enjoying yourself. But bats are put into separate bags, and those bags will contain all the droppings produced that morning while we're processing them. Thus, we have the opportunity of comparing the amounts eaten and the diets of individual bats, and this is invaluable. It shows a completely new, sheds completely new light on what individual bats are doing, which is what you need to do in ecology, because just like humans, not all bats do the same things all the time. Each bat's pellets are counted and weighed after drying. And here you can see that clearly individual bats are eating different prey and very different amounts. All shown were from lactating mothers. If you look at the top, we've got code numbers. Those code numbers are my IDs. Female with a plus is a lactating female. This one here, bottom left shows 41 droppings weighing 140 milligrams, and it applies to the bottom left corner of the plastic box. If you look in the middle there, we've got a bat which hasn't done very well. It's got brown droppings and there aren't many of them. That was number 12450, which was a very old female, and she had 13 droppings weighing only 55 milligrams. But we've got bats here that have eaten 200 or more milligrams of food. So we can pinpoint the successful animals and the duffers. The key message from this study is that whatever habitat and or land management systems are implemented, weather conditions are crucial to foraging success in summer just as much as they are in winter. They may well forage at dusk and eat very little at dawn. And if you get torrential rain that starts at dusk and goes through to dawn, no bat will go out and eat anything. And this happened on one occasion this summer. We've had a summer of extremes, which you might expect with climate change. But when you look at who's eating most, it's the lactating females which eat the most by far, and then the subadult females next, and non-breeding ones least. If a bat has a year off, it will eat very little. So what the bats are doing is matching the amount they eat with the physiological needs they have at the time. And the most stressful thing that a female can do is rear a successful female offspring. Cold, that is less than 6 centigrade, is the most damaging, but that's not normal at this time of year, but it will happen at other, in June, certainly. Wet and windy weather can be almost as harmful. I know it has an impact on pup growth. The next diet research report was concentrated on winter. But you can't radio track bats in the winter. If you do, most of the time you're sat there doing nothing and they're doing the same. So really you've got to do it from droppings which were collected in bags that were bagged up in the winter when we were catching big colonies. We did this in a whole range of places around Bristol, Wiltshire and so on. And we related the extent of foraging to the different age and sex groups. And we identified the main insects which were involved and looked at the variation in the various colonies. And what we found was that the best place by far was the Forest of Dean, because their habitat and their land management processes were highly suitable. The Forest of Dean had a system which protected the grazing rights of people called badgers who had inalienable rights to graze their sheep, and thousands of them were in the forest, almost like wild animals, and they died as well in the woods, and you know, burying beetles were abundant in the area. It was a very, very unique ecosystem, and sadly, a lot of those practices are now disappearing. So which bats forage and when do they do it? We can read Ransome 1968, 71, and then in 2002, the report just then shows that foraging can occur throughout the hibernation period, and I've caught male bats at Christmas that have produced as many drop-ins as some of those summer females did. So it's not only, they're not just eating scraps, they are successfully foraging, and this has an impact on their weight loss. They can forage whenever the daily temperature reaches about 10 centigrade. And what we've got here is evidence from the forest, and you see the wonderful iron ore colours of the rocks as well. So what do bats eat in different seasons? Well, as you can see on the left-hand side, cockchafers, noctuid moths are the top dogs. These are the key prey that the bats will always go for. Okay. Right. Aphodus rufipes is the key prey for the young, and the mothers will eat it if the moths are in short supply. Tipulids I used to classify as a secondary prey, but in actual fact they are highly nutritious, and they are eaten, and possibly next to moths are one of the best insects to have as a backup. In the winter, it's the door beetle. There are 3 of them, Geotrupis species. They fly from August to April, but strangely, the bats don't eat them in August, September, October, and November. They wait until they fattened up and become highly nutritious, and they're then eaten in December through to the following April. The Ophion wasps, and they're the ones that are mostly eaten in the winter, especially the sites around Bristol where there is no winter grazing, and virtually that's what they prefer. And as a last resort, the dung fly, and in some periods, particularly in November, we'll get 100% dung flies being eaten by bats that are bagged up. So what habitats do you need in winter? Well, the answer is the same woodland ecosystems as in summer, only they won't travel as far. Probably 1 kilometre is about the maximum range, and we're guessing a lot. It's very difficult to find that out. The grazers must be present as free ranging herds or flocks. None of the dung animals eat old dung. They go for the fresh stuff, and they either bury it or eat it directly. Dog feces are also used. In the Forest of Dean, there was a very good picture sent to me from Andrea Dern that showed a pile of dog feces that were absolutely crawling with geotroopers. And it was from a dog that they just walked along the path to get to the site to do a count. And when they came back, they could see that these feces were being taken away by the geotroopers. So rather than stick them in plastic bags and throw them in the hedge, it would be better if dog owners put their dogs off the path, and it contributes to the biodiversity for bats. The Ophion wasps are good as long as the moth do well, because they're parasitic, and if the moths fail, so do the Ophions. So if you get bad weather in the summer and you have low moth populations, you'll get very low Ophion supplies, and then the bats are almost forced to nothing but dung flies, and you'll only have those if you've got grazers around. I put maybugs in because maybugs are around. In fact, they can come very early in in April, as well as in May. So they come in periodic periods of swarms, but not necessarily in good numbers every year. A big summary of this was done by Martin Longley in 2003. I expect a lot of you have read it. It certainly was a properly produced and referenced document, and I would recommend that. Woodchester, move on to radio tracking now. Woodchester is within a steep-sided wooded valley. It doesn't have anything like the population that the Forest of Dean has. Dean Hall last summer had 400 adults and 140 babies, which is quite an achievement for such a northern latitude site. A lot of the Woodchester pups, when they grow up, they'll spend their first winter in the Forest of Dean. They'll cross the motorway, presumably underneath some of the ways they get across the motorways. They cross the River Severn at Arlingham, where it's fairly narrow, judging by the radio tracking data, and they go and spend their time with the Forest of Dean bats, and then usually later on they will switch to the more local sites near the mansion. The mansion is the little red blob, whether you can see my cursor, just there. And this is the valley with the lakes. And here we've got large amount, all the purple blobs are increasing urbanization, and we've got major building projects going on down in the valley. Most of the radio tracking data shows that the bats aren't feeding much down in the valley. They are mostly on the hillsides and anywhere where there's grazing animals. When you radio track, you've got to put a tag on a bat, and that tag has to, you shave the fur off and you glue the tag to between the shoulder blades. All right, we're on the last bit. Initial studies were by Stebbings, Jones and Morton, and then Duverger in 1996 tagged mums and babies, and this is one of the babies with the tag on it. It's lovely grey fur which characterises them. The mothers are lovely chestnut brown. He was going to try and find out if whether the mothers and the babies hunted together. And the third study was Rossiter. He published it in his PhD, but also as a joint paper in 2002. And he investigated foraging by related older females from 2 different matrilines. That's Steve with his kit and his off-road Suzuki, which used to manage 35 miles an hour on a good day. Hard work. Laurent found that the mothers left the roosts earlier than the pups and showed no association outside the mansion maternity roost for the whole time that both of them were out. So mothers do not teach babies how to hunt or where to hunt. They do it entirely themselves. And they can do it from 30 days of age. And this is a bat that can live for 30 years. That is an amazing achievement. Steve tagged older female bats that came from the 2 largest matrilines. He had, I think, with one of them, 6 females of different ages, so that he could see what they were doing. And he followed them for 2 to 3 weeks. And the 2 matrilines used separate foraging areas and separate night roosts, but within each matriline, they were associated together, occasionally foraging, sometimes when they perch hunted, but they did share night roosts, so they are a very sociable animal. If you're going to do this sort of work, you really need 2 persons to get fixes. You need data loggers to record the fixes for processing, tape recorder for loads of events to be recorded, a 4-wheel drive vehicle, immense amount of stamina, and poor old Steve suffered from the worst hay fever I've ever seen anyone. There was a time when he could hardly open his eyes, and he deserved a medal for finishing his study. Whoops. Right, I'll leave you with this future slide, as I think I've just about finished my time, so that you can have a look at what were some guesses as to the future. I hope that things will be managed properly after we leave the European Union. I'm pretty certain that the rules will be followed because we set most of their rules anyway. And I would like to end by thanking all those who've helped with this considerable study. Thank you.